called PDM in Pfizer. So we work mostly on the large molecule, including IgG, Fc fusion protein, all kinds of proteins with peptides, and use in vivo, mostly in vivo studies and some in vitro studies to understand the ADME property of these molecules. Before I start, can I get a poll? How many of you have the experience of working on the large molecule, peptides, protein, Fc? Can you raise your hands, please? Okay, thank you. And how many of you have the experience about ADME property, PK properties? Okay, thank you very much. That will give me a good sense about where everybody is, so we can. I'll try to get everybody on the same page by the end of the presentation. So the title is Skin Metabolism of Biotherapeutics as a Limiting Factor for Subcutaneous Bioavailability. I have about 20 slides. I'll try to go slow. I'll start with some background to get everybody on the same page. To start with the introduction of what bioavailability is, introduction of peptide PK, and then overviewing of some of the very quick overview of technologies for peptide half-life extension. And then I will use one case study that we published. I think that's why I'm invited here for, to understand the low sub-Q bioavailability of a peptide. And the example is the oxyntomodulin half-life extension and the problem, what we have done and what we found out from the study. And hopefully it's going to be a fun journey for you, not too boring, but feel free to ask questions. So this is a very quick background introduction of what the bioavailability is. It's a term for pharmacokinetics. It's a fraction of the dose of a drug containing any dosage form that reaches the systemic circulation, which is blood, in unchanged or active form administered through any route. It is known as bioavailability. So sub-Q bioavailability is what we detect in the blood comparing to the IV route, because the drugs injected using IV route of administration have 100% bioavailability, while the others have much less bioavailability because, you know, some of the drugs may not be absorbed from the injection side, either through subcutaneous injection or IP dosing, the interperitoneal dosing, and some of the drug can be degraded, metabolized before reaching the site of action. So, you know, this drug goes in. If it's destroyed at site of absorption, or if it's not absorbed into the blood at all, or if it's metabolized before entering the circulation, the bioavailability would be very low. And in the pharmaceutical industry, it's a big hurdle to predict the subcutaneous bioavailability of all kinds of biotherapeutics in human. And this is a table that listing the problem. You can see that there are all kinds of this therapeutic proteins, some growth hormone, interferon, and then, you know, MABs, monoclonal antibodies, and Fc fusion proteins. They have very different bioavailability across the species, from rat to monkey to human. For example, the interferon beta 1b, the rat bioavailability is 16. In monkey, it has higher range from 12 to 38, and in human, it's even higher from 6 to 60%. So with this, usually what we do is we assume most of the bioavailability should be similar across the species while we're working on the small molecule world, but it's not the case in the larger molecule. So as you see, there's a wide range of bioavailability and high degree of variability. And we don't really have a good animal model to predict and to scale from small species to large species. The preferred bioavailability is 60%, doesn't matter what it is, but usually for therapeutics, it's sub-Q dosing. My talk will focus not from those big molecules, but something that in the middle of small molecule and large molecule world, which is the peptide. And this is a table that I borrowed from Roland and Tozer, which is a very famous book for PK. And you can see when the molecular weight increases, the glomerular sieving coefficient, which is how fast and how much the extent of the drug will be filtered through the kidney, will decrease to almost 0 in IgG. So insulin, which is a kind of a typical peptide with molecular weight of about 6 kD, it has pretty high GSC, 89%. So because of that high and very quick filtering through the kidney, the PK or the half-life of the peptide usually is very short. So peptide with size less than 20 kD, that is the peptide, has half-life ranging from minutes to hours. So it does not make the dosing regimen very easy because we need to dose either BID twice a day or QD at the very best scenario. So if we want to use peptide as a therapeutic, we need to extend their half-life, and there are a lot of ways to extend the peptide half-life. The very typical way is through conjugation. I believe most of you have heard of the pegylation, the PEG growth hormone, the PEG interferon, and there are a lot of others I'm not listing because that's not the focus of the talk today, such as hesylation. This is kind of a starch. It's not toxic. You just conjugate it to the peptide, and because of the bigger size, usually the half-life of peptide is better. And what we focus here is the sustained release. There, as you see, a big list of all kinds of sustained release. There's transdermal patches, polymeric micelles, complexation, injectable depots, such as those in which the drug is embedded in different kinds of matrices, including polymeric matrix, vesicles, microspheres, hydrogels. These are all kinds of, there are a lot of biotech companies doing this. And or a formulation of a reversible water insoluble complex of the protein with divalent metal cations, or the combination of above. And by using the sustained release formulation, it can not only extend the half-life, but also reduce the fluctuation between peak and trough concentration, which is pretty much what we expect. And I use an example here that, if you use a peptide alone, the PK, the concentration drops very fast in this case. After 24 hours, it's almost gone. But if you use a sustained release, it will be slowly released from the depot, and you can detect the drug concentration pretty much after 2 days. So now I switch gear to this case study that we were trying to develop oxyntomodulin, which is a peptide, as a target. And it is the target for type 2 diabetes and obesity. And oxyntomodulin is a 37 amino acid peptide with about 3 to 4 kD, and it is secreted by the L cells of the small intestine. It's derived from, you can see, it's derived from this big gene, pre-pro-glucagon gene, by the PC convertase. And this is the oxyntomodulin that is being spliced. And you can see it's the combination of glucagon with some peptide. It is 8 amino acid octapeptides. It's mostly exists in pancreas for these peptides. And for these peptides, oxyntomodulin, GLP-1 and 2, mostly in brain and gut. So, and you can see this is the sequence of oxyntomodulin with glucagon and octapeptides. It's mostly cleaved by two peptides, DPP4 and NEP, neutral endopeptidase. It's very much conserved. It's homologous 100% between human, mouse, and rat. Because of its instability degraded by these two enzymes, it's not appropriate therapeutic due to the proteolysis and poor PK properties. So how do we develop it as a therapeutic? So Pfizer has used a site-directed mutagenesis, created oxyntomodulin analog, and we call it peptide A here. It is designed to be stable against DPP4 and the NEP. So with this resistant to the proteolysis, it has in vivo half-life of 3 hours in human now versus 10 minutes for the original oxyntomodulin. And its pharmacology effect, which is the short-term weight loss, is observed in human after sub-Q injection of this peptide. But still, 3 hours is pretty short, even for small molecule. So the company developed a formulation, which is the controlled release form for subcutaneous dose, and they were evaluated in the preclinical space, and they found that the CR, the controlled release form, has provided the prolonged exposure compared to the instant release form, which is the IR form. So this, I'm gonna talk quickly about what we have found preclinically. So this is the figure that you've seen to show the extended half-life of the controlled release CR form. But to our surprise, if you look at the half-life extended, but what we lost is the bioavailability. The subcutaneous bioavailability for the instant form was only 15%, and we were trying to shoot for 60%. And the CR form, the controlled release form, was even lower. It's only 9%. So what was the problem? Why do we have such a small bioavailability? And how can we increase once we know the mechanism? And by the way, the following work, the study was published in the Journal of Bioequivalence and Bioavailability in 2012, just in case anybody is interested. So why the low bioavailability? There are a couple of possibilities. First, we want to know, where does the drug go? Is it because it's not absorbed through the skin and still reside in the injection site, or it's degraded in the skin or somewhere else before entering in the system circulation? So what we were going to do is to do a dose recovery study to see where exactly the drug is, and then we follow up with some in vitro stability because we thought, there was some degradation in the skin. We used some in vitro stability study to prove the skin degradation and follow up with the metabolite investigation, both from in vitro and in vivo. Before I go to the radio-labeled study that we conducted, just so you know what we do, we usually label the drug with a radioisotope, and I-125 is the most common one we use. After the drug is labeled and dosed to the animals, serum or tissue or plasma is collected, and we will count the total CPM, total radioactivity of the sample. And we also will use trichloric acid, which is a kind of acid that can precipitate the protein portion, and then we separate the supernatant and this precipitable portion. The precipitable portion, which is here, we consider it's related to the intact drug, and the precipitate, the supernatant, is considered as the degradants. So that's how we can see, follow the PK of the intact form. We can also follow up If there's any question, we follow up with SDS page gel or HPLC to have a finer investigation of the radioactivity. Are they intact really or metabolized? So we did this dose recovery study of the I-125 labeled peptide A. And we gave two route, IV and sub-Q, different doses. And then we collected serum, tissue, all kinds of a panel of tissues, including brain, liver, pancreas, kidney, whatever you can name it, and urine and feces. And then at the end of the study, we count all the radioactivity and add them up and see if we can get all the radioactivity recovered. And then we can know where the radioactivity mostly concentrated. So, so let's focus on this lower right figure. So this is the radioactivity or converted to concentration across the time frame where, for plasma. In this case, it's the open circle, and also the radioactivity after instant release form at this injection site on the skin. And the bottom one is for the controlled release form. And when we see the parallel of plasma and a tissue, we think it's pretty much reaching the steady state, meaning there's no accumulation in this tissue. So it was actually pretty good for both IR form and the CR controlled release form that they reach the steady state. There's not much accumulated into the injection site. The only thing is that for the controlled release form, the radioactivity keeps going up and then goes down, meaning, for 3 days, 72 hours, meaning that, the absorption is pretty much slower than the instant release form, which is expected. That's what we want. So now let's look at the table for the percent dose recovery. You can see after both IV and sub-Q injection, the percent dose recovery of the radioactivity was pretty good. This is very good recovery if we've done so many studies, if it's above 85%, it's pretty good recovery. So we can see all the radioactivity, and we can see most of the radioactivity resides in the urine, indicating that most of these drugs are either metabolized or secreted through the kidney directly. So we didn't lose much drugs, and indicating that the radio-labeled dose was well absorbed from the injection site. So if it's totally absorbed and the bioavailability is still low, something must be degrading this peptide before going into the injection. The first thing we studied was the injection site. Was the peptide degraded in the skin? So we followed up with in vitro study, still using the radio-labeled peptide because, with the radio labeling, the property of I-125 makes, analysis very, very simple. We just take the sample, put in the scintillation counter and count, and we get the concentration right there. So what we did was we call it plunk and dunk because this is a very fast and dirty experiment. It does not mean to give us very accurate value, but just give us a rough idea of is there a lot of metabolism or no metabolism. So we cut the skin 1 inch in diameter, freshly cut from rat after sacrificing, or we boil another skin sample using as a negative control because most of the protease is supposed to be inactivated after boiling. And we incubate these skin samples with peptide at 37 degrees. So after different time points, we take out the incubation solution and read the total counts and read the TCA soluble counts. The TCA soluble counts, as I described previously, is indicating the degree of metabolism. So the blue is the freshly cut skin sample incubation, and the red is the boiled skin sample incubation. And the dotted line is the soluble counts indicating the degradants. When we look at the negative control, which is the boiled skin, we didn't see much change of all the radioactivity in the total counts and not much in the soluble counts. However, we were very excited to see that when we incubated the peptide with the fresh skin, we see very much increase of the soluble counts, indicating even in an in vitro situation with only 1 inch of diameter skin sample for only like an hour and a half, almost 80% of the peptide was degraded. It kind of validates our hypothesis. So after we knew that the skin was the major site for metabolism of this peptide, we followed up with the metabolite identification. Again, we used the samples from the previous experiment and ran through LC, HPLC. The LC is coupled with the chromatography. So this is the top panel is the skin sample after 1.5 hour incubation with the boiled skin. And you see most of the radioactivity relate to the intact peptide. And after 1.5 hour in vitro incubation with the fresh skin, we see the intact peptide decreased and a big peak, much smaller, appeared. And we think that's the metabolite we were looking at. And then we also dig out the in vivo study that we did from the freezer and took the skin out and then did a lot of extraction from the skin. From the 30 minutes after sub-Q injection skin sample, we were able to detect the same metabolite from in vivo. That was another further step of confirming, there is metabolism and the metabolite was detected. So we were able to detect the sub-Q. We followed up with almost all the sample that we collected from the in vivo study. It was interesting, after IV dosing in the plasma samples, we were able to detect the peptide itself after 3 hours. And this is the peak front, is not really any signal related to any drugs, and we couldn't detect any metabolites from the IV dosing. And this is the plasma samples after the sub-Q dosing. After 1 hour of post sub-Q dosing, we were able to detect the same metabolite. There were still a lot of follow-up studies, but I just want to stop here because this was kind of a complete story. So just to conclude, the dose recovery data from radio-labeled PK studies in rats revealed that the peptide-related radioactivity was completely absorbed. The in vitro study showed the increased formation of degradants during the incubation with freshly cut rat skin, but not with the boiled skin. And we were able to find this major metabolite using the HPLC radiochromatography. And the metabolite peak was also observed in both the subcutaneous administration, the skin sample, as well as the plasma samples. So the results from this study, that's the take-home message, demonstrated that while the controlled release form can prolong the systematic half-life of peptide, which is usually what we desired, but the metabolism of the peptide in skin kind of offset its prolonged residence because, it's just the available drug to be absorbed is decreased. So caution needs to be taken to balance the increase of half-life and the decrease of exposure while choosing a right formulation. Before I end up, I want to acknowledge a lot of people that worked on this project. a lot of them are now in different companies across, all over the world. But so this core group was helping me with all the radio-labeled in vivo and in vitro studies. And Kathy used the HPLC, and a lot of group in here helped me with the formulation, and a lot of our comparative medicine colleagues help us with the in vivo study, dose administration. So with that, I want to let you enjoy the picture of our Pfizer site at Andover, Massachusetts. We have a small pond that we can walk around during lunchtime, and there's little fish and turtles in there, and Canadian geese wander around. It's a pretty beautiful scene, and welcome to Boston whenever you have chance. Thank you very much for your attendance. Thank you.